Auxin, a cornerstone in the world of plant biology, represents one of the most influential classes of plant hormones, orchestrating a myriad of developmental and physiological processes that underpin plant life. Since its discovery in the early 20th century, Auxin has fascinated botanists, molecular biologists, and agronomists alike due to its central role in shaping plant architecture, directing growth, and mediating responses to environmental stimuli. The study of auxin biology is not merely an academic pursuit. It holds profound implications for agriculture, ecology, and our broader understanding of how life adapts, survives, and thrives on Earth. At its core, auxin refers to a group of structurally related compounds, with indole 3-acetic acid, IAA, being the principal naturally occurring form. Auxins are synthesized predominantly in young, actively growing tissues, such as shoot apices, young leaves, and developing seeds. Their distribution within the plant is tightly regulated, both spatially and temporally, ensuring that growth and development are coordinated with remarkable precision. This regulation is achieved through a complex interplay of biosynthesis, transport, conjugation, and degradation mechanisms, all of which are subject to modulation by internal and external cues. The history of auxin research is a testament to the ingenuity of early plant scientists. In the late 19th and early 20th centuries, experiments by Charles Darwin and his son Francis laid the groundwork for understanding plant tropisms, directional growth responses to environmental stimuli, such as light, phototropism, and gravity, gravitropism. These pioneering studies suggested the existence of a mobile chemical signal that could influence growth at a distance from its site of perception. Subsequent work by Fritz Wendt, who isolated and characterized auxin activity in oat coal optiles, provided the first concrete evidence for such a substance, ushering in the modern era of plant hormone research. The biosynthesis of IAA, the predominant auxin, involves multiple pathways, reflecting the evolutionary importance and complexity of this hormone. The primary route in most plants is the tryptophan-dependent pathway, wherein the amino acid tryptophan is converted into IAA through several enzymatic steps. Key enzymes such as tryptophan aminotransferases and yucca flavin-containing monooxygenases catalyze critical reactions, and their expression is finely tuned according to developmental stage and environmental conditions. Alternative, tryptophan-independent pathways also exist, though their significance varies among species and contexts. Once synthesized, auxin must be distributed to target tissues, a process that relies on both passive diffusion and active carrier-mediated transport. The latter, known as polar auxin transport, is essential for establishing and maintaining auxin gradients that dictate developmental patterning. Specialized transporter proteins, including the pin-formed PIN family, AUX1, ALAX influx carriers, and P-glycoproteins, coordinate the directional movement of auxin across cell membranes. The asymmetric localization of these transporters within cells enables the plant to direct auxin flow, creating zones of high and low auxin concentration that act as positional cues during organogenesis, vascular differentiation, and other developmental processes. Auxin perception and signaling constitute another layer of regulatory complexity. For decades, the molecular nature of the auxin receptor was elusive, but breakthroughs in the early 21st century identified the transport inhibitor response, yin auxin signaling F-box, TIR1, AFB family of proteins as bona fide auxin receptors. These F-box proteins, as part of the SCF-RTIR1 AFB ubiquitin ligase complex, bind auxin and promote the ubiquitination and subsequent degradation of AUX-IAA repressor proteins. This degradation releases the inhibition on Auxin response factor, ARF, transcription factors, allowing them to modulate the expression of auxin-responsive genes. Through this elegant mechanism, auxin rapidly alters gene expression profiles, enabling plants to adapt their growth and development to changing conditions. The physiological effects of auxin are vast and diverse, reflecting its pleiotropic nature. One of the earliest and most studied auxin responses is cell elongation, particularly in stems and coleoptiles. Auxin stimulates proton pumps in the plasma membrane, acidifying the cell wall and activating enzymes that loosen the wall structure. This acid growth hypothesis explains how auxin facilitates cell expansion, contributing to tropic responses and overall organ growth. Beyond elongation, auxin regulates cell division, differentiation, and pattern formation. It is indispensable for embryogenesis, root and shoot branching, vascular tissue development, and the formation of lateral organs such as leaves and flowers. Auxin's role in apical dominance, a phenomenon where the main shoot suppresses the growth of lateral buds, has been a classic subject of study. High auxin levels in the shoot apex inhibit the outgrowth of axillary buds, maintaining the hierarchical structure of the plant. When the apex is removed, auxin levels decline locally, allowing lateral buds to activate and grow. This simple yet powerful mechanism illustrates how auxin integrates developmental signals to optimize plant form and resource allocation. In roots, auxin governs both primary root elongation and lateral root initiation. Gradients of auxin concentration determine the sites where new lateral roots will emerge, while localized maxima stimulate the necessary cell divisions and differentiation. Moreover, auxin interacts with other hormones, such as cytokinins, abscisic acid, and ethylene, to fine-tune root architecture in response to environmental cues like nutrient availability and water status. Tropic responses, such as phototropism and gravitropism, offer vivid demonstrations of auxin's role as a mediator between environmental perception and developmental outcome. In phototropism, for instance, differential distribution of auxin to the shaded side of the stem causes those cells to elongate more, bending the plant toward the light. Similarly, in gravitropism, auxin redistributes in response to gravity, promoting differential growth that reorients roots downward and shoots upward. These adaptive responses enable plants to optimize their exposure to light and anchorage in the soil, conferring survival advantages in dynamic environments. Auxin also plays critical roles in reproductive development. During flower formation, auxin gradients guide the initiation and patterning of floral organs. After fertilization, auxin produced in developing seeds stimulates fruit growth and maturation, coordinating the intricate dance between maternal tissues and progeny. In some species, application of exogenous auxin can induce parthenocarpic, seedless, fruit development, a trait of considerable agricultural value. The interplay between auxin and other plant hormones exemplifies the sophisticated regulatory networks that govern plant biology. Auxin and cytokinin often act antagonistically in controlling organogenesis and meristem maintenance, while interactions with gibberellins, brasinosteroids, and ethylene further modulate growth responses. These hormonal crosstalks ensure that plant development is robust yet flexible, capable of integrating multiple signals to achieve optimal outcomes. Recent advances in molecular genetics, genomics, and imaging technologies have revolutionized our understanding of auxin biology. The identification of genes involved in auxin biosynthesis, transport, and signaling has enabled researchers to dissect the underlying mechanisms with unprecedented detail. Mutant analyses, reporter gene constructs, and live cell imaging have revealed the dynamic patterns of auxin distribution and response during development. Furthermore, systems biology approaches are beginning to unravel the complex gene regulatory networks downstream of auxin signaling, shedding light on how a single hormone can exert such diverse effects. Despite these advances, many fundamental questions remain. The precise mechanisms by which auxin gradients are established and maintained, especially in complex tissues, continue to be an area of intense investigation. The diversity of auxin receptors and their context-specific functions suggest layers of regulation that are only beginning to be understood. Additionally, the interplay between auxin signaling and cellular metabolism, epigenetic modifications, and environmental sensing represents fertile ground for future research. The practical applications of auxin biology are equally significant. Synthetic auxins, such as 2,4-dichlorophenolactic acid, 2,4-D, have been widely used as herbicides, rooting agents, and in tissue culture protocols for plant propagation. Understanding auxin dynamics has informed breeding strategies for improved crop architecture, yield, and stress resilience. In forestry, horticulture, and biotechnology, manipulation of auxin pathways offers avenues for optimizing plant growth, controlling invasive species, and engineering novel traits. Auxin research also intersects with pressing global challenges. As climate change alters environmental conditions, the ability of plants to adapt their growth and development becomes increasingly important for food security and ecosystem stability. Insights into auxin-mediated responses to drought, salinity, temperature extremes, and pathogens can inform strategies for developing resilient crops and sustainable agricultural practices. Moreover, a deeper appreciation of auxin biology enhances our understanding of plant-environment interactions, biodiversity, and the evolutionary innovations that have enabled plants to colonize virtually every terrestrial habitat. From a broader perspective, the study of auxin exemplifies the beauty and complexity of biological regulation. A single class of molecules, through intricate networks of synthesis, transport, perception, and response, orchestrates the form and function of the plant kingdom. Auxin's story is one of discovery, curiosity, and the relentless pursuit of knowledge, a testament to the power of scientific inquiry to illuminate the fundamental processes of life. As we look to the future, the integration of auxin biology with emerging fields such as synthetic biology, computational modeling, and precision agriculture promises to unlock new frontiers. By harnessing the principles of auxin action, we may engineer plants with tailored architectures, enhanced productivity, and greater resilience to environmental stressors. At the same time, ongoing research continues to reveal the subtlety and sophistication of auxin-mediated regulation, reminding us that even the most familiar aspects of nature harbor mysteries yet to be unraveled. In summary, auxin stands as a central pillar of plant biology, shaping the growth, development, and adaptability of plants in ways both visible and hidden. Its discovery and subsequent study have transformed our understanding of how plants function, respond to their environment, and interact with other organisms. As research progresses, auxin will undoubtedly remain at the forefront of plant science, offering insights not only into the workings of the green world, but also into the universal principles that govern living systems. Through the lens of auxin biology, we glimpse the intricate choreography of life itself, a dance of molecules, cells, and organisms, perpetually adapting and evolving in the ceaseless quest for survival and flourishing.